Heme portion of hemoglobin is synthesized from glycine and succinyl-CoA. Heme is synthesized by cells of erythroid series in red bone marrow. Hemoglobin starts appearing in the developing of RBC at the intermediate normoblastic stage. These are the different steps of synthesis of hemoglobin. 2-succinyl-CoA and 2-glycine forms the pyrrole ring. 4 pyrroles, they are going to form protoporphyrin ring, and protoporphyrin with iron, it is going to lead to formation of heme. Heme and polypeptide, depending on the alpha or beta chain, they are going to form hemoglobin chain, and 2 alpha chains and 2 beta chains together, they will form hemoglobin A. Hemoglobin is a globular structure made up of 4 subunits. Each subunit contains heme moiety conjugated to a polypeptide which is called as globin. Heme is an iron-containing porphyrin derivative. There are 2 pairs of polypeptides in each hemoglobin molecule. Hemoglobin A consists of 2 types of polypeptides, that is alpha and beta, and one hemoglobin molecule can combine with 4 molecules of oxygen or 8 atoms of oxygen. This is the structure of hemoglobin, which is made up of the heme structure. So this is the structure of the heme, and these are the 4 polypeptides associated with each hemoglobin. In each hemoglobin, you have 2 chains, 2 alpha chains and 2 beta chains. Normal values: males have 14 to 18 grams per cent of hemoglobin content in their blood, whereas females little less, average is around 14 gram per cent. There can be different types of hemoglobin. The adult hemoglobin, there are 2 types, hemoglobin A and hemoglobin A2, where the hemoglobin A has alpha 2 and beta chains, whereas hemoglobin A2 has alpha 2 and delta 2 chains. Whereas fetal hemoglobin has alpha 2 and gamma 2 beta chains, and functionally fetal hemoglobin is different from adult hemoglobin, that it has a more affinity towards the oxygen. Some of the abnormal varieties are seen in hemoglobinopathies, and these are hemoglobin S. In the hemoglobin S, in the beta chain, glutamine at the 6th position is replaced by valine. When HbS is reduced, it precipitates into crystals within the RBC, leading to sickling of RBCs, and it occurs whenever the RBCs are exposed to the hypoxia. And sickle-shaped RBCs are more fragile, resulting in sickle cell anemia. The other hemoglobinopathies are hemoglobin C, hemoglobin E, and hemoglobin I. Thalassemias. Both alpha and beta chains are present in these diseases and are normal in structure, but produced in reduced amounts, or sometimes they are absent. There are variants in thalassemia. It can be major beta thalassemia or minor beta thalassemia. Major beta thalassemia is also called as Cooley's anemia or Mediterranean anemia. It's a less common. There will be total absence of chain synthesis. It has a homozygous transmission, and victim usually dies because of the major beta thalassemia, because of excessive of hemolysis. Minor variant is more common, and it occurs because of the partial synthesis of beta chain, and it transmitted heterozygous pattern. So these are the variants of thalassemia. It can be beta thalassemia because of the absence or reduced beta chain synthesis, or it can be because of the reduced alpha chain synthesis, which is rare. Beta thalassemia can be major and minor. Minor is more common because of it occurs because of the partial production of the